Welcome, everyone. Just giving a minute here for people to join. All right, I will get started with my part of the introduction here. Thank you so much for taking time this morning to be with us and tune in. We're all excited to be here and share some really great information with you all. I'm Allie Kajowski. I'm the Director of Outreach at the American Society of Gene and Cell Therapy. I've been working on our patient education efforts for over 7 years. More recently, I met Jainu at one of our events in May. He shared with me that there was a lot going on to develop a gene therapy for CDKL5. So since then, we reconnected and put together this webinar for the community. And I just want to start by introducing who our two speakers are today. So some of you may know Jainu Jogani, but he's a proud father of Raina, his 7-year-old daughter, who lives with CDKL5 deficiency disorder. With this, he brings a unique blend of personal passion and also some professional expertise. He has a background in biomedical engineering and has received orphan drug designation from the US FDA for a small molecule drug targeting CDKL5. He is also the co-founder of Child's Cure Genetic Research and the founder of CDKL5 South Asia. So we'll hear from him later. And then also bringing her scientific expertise, we're joined by Dr. Rachel Bailey. She's an assistant professor at the Center for Alzheimer's and Neurodegenerative Diseases and Department of Pediatrics at the University of Texas Southwestern Medical Center. Dr. Bailey's research focuses on the development of gene therapies for neurological disorders and helping get these treatments to human clinical trials. We appreciate the questions that everyone submitted in advance. We'll hopefully address many of these along the way. You're also welcome to submit questions again. We hope to have a little bit of time for Q&A at the end. And then I think we'll get started. I'm just going to share a little more about ASGCT. You can do next slide. So we're the primary professional membership organization for researchers, clinicians, and advocates working in the field of gene and cell therapy. And we're working to advance the knowledge and awareness of these therapies and hopefully expand access to treatments. As you can see, we do a lot of different programs and events throughout the year. One of the programs we do is patient outreach. And that provides useful resources and opportunities like this to make sure the patient community understands gene and cell therapy, clinical trials, and all that goes into this. Next slide. I want to be sure you're all aware, in case this is new to you or not new, or maybe you have people you'd like to share this information with. We do have a whole website of resources on gene therapy. If you go to the QR code there, you'll see just a short animated video on what is gene therapy, and we have a whole website to explore that can help expand your understanding of this topic, and it's all free to share and use. We also have community quick takes. These are a little bit deeper dive, but they're just short 20-minute Q&A on different topics, and you might have heard of different things like immune responses to AAV or wanting to prepare for the unknowns of a clinical trial. So we would welcome you to explore that as well. And then lastly, if you'd like to engage with us in March, we have 2 half days, a virtual summit. It's called Empowering Patients. And I saw a lot of the questions that were submitted, and we try to answer a lot of these throughout those types of events as well. So we would really welcome you and your family to join us for Empowering Patients, and stay tuned. We have a newsletter that you could subscribe to, and that's how you can just stay up to date on news in the rare disease space, as well as resources we have going out and other events available. So I will pass it over. Again, we're honored to be here, and thanks for getting us started, Dr. Bailey.
All right, well, thank you so much for the introduction and the information on ASGCT. And then for today, we just want to kind of level set for why we're here and what our goals are. And so the goal is absolutely to demystify the science behind gene therapy so that we can empower decision making on the behalf of the community and patient advocates. And so our specific objectives are to define different types of genetic therapies, overview the clinical trial readiness and design process, and then also to set some expectations of clinical trial participation. So before we get into that, I just want to show my disclosures. So while I have intellectual property for gene therapy approaches that have been licensed by different biopharmaceutical companies, I actually do not work on CDKL5 deficiency disorder. I am here today on behalf of the ASGCT Speakers Bureau to provide background information on what is gene therapy and is not a specific endorsement of any given approach for gene therapy for CDKL5. So let's first just do some brief background for what is CDKL5 deficiency disorder, or CDD. So CDD is caused by genetic defects in the cyclin-dependent kinase like 5 or CDKL5 gene. The CDKL5 gene provides instructions for making proteins that are essential for normal brain and neuron development. This X-linked neurodevelopmental disorder arises from deletions, truncations, or missense variants that cause the loss of CDKL5 protein function. Loss of normal CDKL5 protein activity affects the proper formation and maintenance of brain circuits. So while CDKL5 is most highly expressed in the brain, it is also found in cells and tissues throughout the rest of the body, although we're still learning what its function there may be. So, as many of you unfortunately know, patients with CDD experience very severe symptoms. These include early onset seizures that often begin in the first few weeks of life, severe global developmental delays, sleep disorders, gastrointestinal dysfunction, visual problems, and breathing abnormalities. This is a disease of extremely high unmet medical need. There are currently no effective FDA-approved treatments available for patients, and seizures are generally not well controlled with anti-epileptic drugs. Importantly, patients typically require 24/7 care due to the severity of the symptoms. So I want to briefly discuss what is the normal function of CDKL5 protein in the brain. So CDKL5 is a protein that plays an important role in helping brain cells grow and make proper connections. It works a bit like a traffic controller and construction manager for the cell's internal scaffolding, the structures that help neurons extend their long branches and form communication points called synapses. So research shows that CDKL5 helps neurons grow long wires that send signals to other cells. It also helps build branches and dendrites, which are the tree-like structures that receive signals. CDKL5 also helps to form and strengthen synapses, which is where neurons exchange information. So it does this by keeping the cell's internal framework, called the microtubules, which are shown here in green, flexible and able to move into new growing areas. CDKL5 also helps organize proteins at the synapse so that communication between neurons is fast and reliable. When CDKL5 is missing or not working properly, though, these microtubules can become too rigid. As a result, neuron growth may be stunted, connections between cells fewer or weaker, and overall brain development can be disrupted. Scientists are still working to understand all of the pathways involved and how the brain tries to compensate when CDKL5 is not present. Understanding that CDD results primarily from the loss of CDKL5 function directs us towards therapeutic approaches aimed at restoring, replacing, or enhancing CDKL5 activity in the brain. So how can we do that? To get everyone on the same page, I briefly want to review what is genetic material and where is it located within our cells. Genetic material most commonly refers to DNA or RNA. DNA stores the genetic information so the cell can continue building proteins. It is permanent and stored in the nucleus in a form called chromosomes. DNA is made up of two strands and is much larger than RNA. Genes are specific sections of DNA that encode sequences, which are a set of instructions, for making proteins. Other sections of DNA can control when, where, and how much of the protein is made. RNA is a copy of the genetic information contained within DNA. While DNA is the permanent storage format of genetic material, RNA molecules help turn those instructions into proteins. After RNA is made within the nucleus of the cell, most RNA moves to the cytoplasm, which is the fluid space between the nucleus and the cell membrane. There are many different types of RNA. Some RNAs carry the instructions for making proteins, such as CDKL5, while other types of RNA can prevent proteins from being made. Importantly, RNA molecules are only active for a limited period of time in the cell. Over time, they get degraded or broken down by the cell, and new RNA needs to be made using the DNA. So gene therapies are biological drugs where DNA or RNA is being delivered to affect a cellular function. There are many types of gene therapies, and some of the most common ones that can be classified as either DNA, RNA, or antisense oligonucleotide therapies, and these can be separated into different approaches. Gene addition is the use of DNA that codes for the production of a specific RNA or protein to treat a disorder. To have a therapeutic effect, the DNA must be delivered to the nucleus of a cell, where it can then be used by the cell to affect protein expression. This gene will now live in the nucleus, which gives it a greater chance of being permanent and is typically only given one time. Sometimes the therapy is designed for the new gene to insert itself into the main DNA storage, while other times it will stay next to the main DNA storage, like an extra set of instructions. Gene silencing is where the delivered genetic material prevents or inhibits the activity of a gene that is already present in a cell. Gene silencing often decreases the amount of a specific protein being made. Gene editing corrects pieces of DNA by changing or deleting the information within the affected individual's gene. Genetic material is sent to directly edit or change pieces of DNA already located within a cell to correct the protein being made by that DNA. Gene editing uses technology that is highly precise to make these types of permanent changes. While this technology is exciting and is actively being explored for a number of disorders, there are many challenges that exist for applying it to brain disorders that scientists are actively working to overcome. And lastly, genetically modified cell therapy involves removing a patient's cells and modifying them in a lab so they can deliver helpful proteins when returned to the body. So as CDD is a brain disorder caused by the loss of normal CDKL5 function, a reasonable therapeutic approach could be gene addition. Our bodies, though, are developed to prevent the introduction of foreign DNA and RNA into our cells. So vectors are typically used to deliver genetic material such as DNA into a cell. There are many types of vectors, and one of the most common approaches is to use a virus. This is because viruses are very good at entering cells. We can engineer viruses to remove their DNA and use the virus shell as a delivery vehicle for a therapeutic gene. There are many different types of viruses that are used for gene therapy, and each has their own benefits and limitations. So here I am showing 3 of the most commonly used virus vectors. Lentivirus is typically used for ex vivo approaches to genetically modify patient cells in the lab before they are then returned to the body. Adenovirus is typically used for cancer treatments. For in vivo gene therapies, though, the most commonly used virus is adeno-associated virus, or AAV. AAV is a small virus that depends on the co-infection of a helper virus for replication, and it's pictured here. As such, AAV alone is non-pathogenic. AAV has a small genome that can be easily manipulated to remove and replace the viral repping cap genes with a transgene of interest, which in turn eliminates viral replication. Importantly, AAV-delivered transgene expression is essentially permanent in non-dividing cells, such as neurons, following a single dose. Multiple serotypes exist with distinct tissue tropism, and of these, AAV9 is the current gold standard for targeting the brain and spinal cord. With the number of clinical trials run thus far and 7 approved AAV-based treatments worldwide, AAV vectors have emerged as viable delivery method for in vivo gene therapy. For neurological disorders, treating the brain is difficult because of the presence of the blood-brain barrier that prevents most drugs from being able to enter the brain through non-invasive delivery methods. Use of AAV is one way to overcome this. The most common way to deliver AAV is through direct brain injections. In preclinical studies in mice, fairly widespread expression of AAV can be achieved from a single direct brain injection. However, when we translate that to the human brain, the spread is limited to about the size of a marble. So while direct brain injections can be very effective in targeting specific brain regions, it is not ideal for widespread gene delivery in the human brain. The least invasive method is an intravenous or IV injection into the blood, but then the AAV serotype must be capable of crossing the blood-brain barrier to infect cells in the brain and spinal cord. AAV9 is one such serotype that's been shown to cross the blood-brain barrier across multiple species. Brain expression of AAV9 is dose-dependent in that the more viral particles that are injected, the greater the distribution of the virus in the CNS. There are several barriers to translating IV injections of AAV into humans, including the high dose of virus required to achieve widespread brain and spinal cord transduction, which results in high manufacturing costs, as well as resulting in high transduction of peripheral organs and inhibition of transductions by anti-AAV neutralization antibodies, or antibodies that will recognize and bind the virus to destroy it. So to address these potential issues, AAV9 can be directly injected into the cerebral spinal fluid. This can be done by injecting into the brain ventricles, which is an ICV injection, or by an intrathecal injection into the fluid that surrounds the spinal cord. This can be done as an intrathecal lumbar puncture injection below the spinal cord, or as an intracisterna magna or ICM injection into the space that is right underneath the cerebellum. When AAV is delivered into the spinal fluid, the strongest effect is typically near where it's given, and it slowly gets weaker the farther it travels. Importantly, following injection into the spinal fluid, AAV9 escapes into the bloodstream to then also go into peripheral tissues and organs, although it is greatly reduced or peripheral organ distribution as compared to an IV injection. So having discussed what are gene therapies, I now want to talk about how we develop novel first-in-human drugs. Here I am showing the traditional roadmap for the development of a drug. During the preclinical period, the drug itself must be designed, developed, and tested for safety and efficacy using cell and/or animal models. If preclinical testing supports that drug is safe and likely beneficial, it then moves forward to clinical testing. An investigational new drug, or IND document, is submitted to the FDA to request permission to start a clinical trial. Once approved, the traditional clinical trial design is to have a phase 1 trial that assesses safety and a small number of patients, which, if positive, then moves to phase 2 testing in a greater number of patients for additional safety testing and initial efficacy testing. If promising, a phase 3 trial in a larger number of patients is then initiated to test for efficacy or the benefit of the drug. If a quantifiable benefit is achieved in a modality specific to the disease, then a new drug application can be submitted to the FDA for review and approval. And even after approval, there's a phase 4 trial in which the benefit and safety of a drug is surveyed post-marketing. So when it comes to clinical trial testing, important considerations for rare diseases must be made to account for the number of patients that exist and are able to participate in clinical trials. For a rare disease, it may not be feasible to follow the traditional 1, 2, 3 trial design. As such, the FDA does allow a single registration enabling clinical trial where safety and efficacy are tested in parallel in a smaller number of patients. For that trial design to be successful, though, the natural course of the disease must be known and outcome measurements and biomarkers identified prior to the start of the trial. These outcome measurements and biomarkers must be quantifiable and associated with disease progression, so that if you then give a potential therapeutic, you can measure the changes in these parameters over time to determine if the drug has halted disease progression and/or restored normal function or healthy levels of biomarkers. For a rare disease, for the phase of the drug development to be successful, it necessitates the participation of patients. And so gene therapy has already changed what's possible for people with rare diseases. By giving the body a working copy of a missing or faulty gene, several treatments have shown life-changing results, helping children with spinal muscular atrophy sit, stand, and even walk, restoring vision for people born with certain forms of blindness, and improving immune function in disorders once considered fatal. These successes showed that treating the root genetic cause can offer lasting benefits and give real hope to families affected by rare conditions. While the use of gene therapy is promising for many disorders, there are challenges to be considered. One is time. Even once a drug is developed and clinical trial testing starts, it takes years for clinical testing to be completed in a safe and rigorous manner to support the FDA approving a new drug for the treatment of patients. Additionally, there are high costs that go into the work it takes to prepare a clinical trial, and in particular to manufacture viral vectors and ensure it is carried out at the highest standard and ensuring safety for participants. Most gene therapies are developed for disorders caused by single gene variants and may not be applicable to all patients with that disorder. As such, different approaches are often needed. Finally, although there are now many FDA-approved gene therapies, accuracy and efficiency of expressing a gene in the right tissue at the right level and for the right amount of time remains a challenge. In addition to the challenges I just mentioned, there are risks of gene therapy to be considered. The first is regarding safety and immunity. People may be naturally infected with viruses such as AAV over their lifetime, which can cause an immune response and may exclude patients from being able to receive a viral-based therapy. When given a gene therapy, a patient may also have an immune response to either the vector that was used for delivery or the genetic changes that are made as a consequence of the gene therapy. As such, immune suppression is typically used for delivery of an in vivo viral-based gene therapy, and patients are typically monitored closely for an immune reaction that in the weeks following delivery. It also needs to be clear that therapies being studied in clinical trials are not a guaranteed cure and cannot guarantee beneficial results. There is always a chance that the investigational new treatment may not work or may have unexpected side effects. Participating in an AAV clinical trial may also prevent future participation in other trials or from additional types of treatments. Finally, gene therapy can be an alteration for a lifetime, so people should be aware that there could be long-term effects, both good or bad, that are unknown at the time. That is why gene therapies are monitored in a person for years following treatment. So the last point I want to touch on is the role of families and patient advocates in drug development for rare disorders. For a drug to be developed for a disease, there must be the need. If there is a large number of individuals affected, then industry will often lead drug development efforts as there is a high financial return with a successful drug. For a rare disease, though, this burden often falls on families with affected children to then raise awareness and provide the initial funding and drive for research. In addition to funding, drug development requires use of either cell or animal models that may not exist. This is where foundations play an important role in helping to identify the best pre-existing models for researchers to use if available, supporting the development of new models as needed, and helping to collect patient cells to develop cell-based models. They typically also need to engage with researchers in the field that are the experts in the development of gene therapies to design the drug and carry out the preclinical studies. If preclinical studies are promising, it is then important that advocates help support clinical trial readiness. This comes in the form of determining biomarkers and outcome measurements through natural history studies, and being able to identify patients eligible to participate in a clinical trial through the patient registry. As part of the regulatory interactions with the FDA, the sponsor of a clinical trial must provide evidence of a patient population that can participate in clinical trial. This is often a challenge for rare disease, but it's possible by working with strong and active foundations to make it happen. So with that, I actually now want to turn over to Janu Jagani. He, again, as Ali had mentioned, is the father of a 7-year-old daughter that is impacted by CDD, Rayanna, and he has a background in biomedical and is now working in robotics industry. And to reiterate, he has orphan drug designation by the US FDA for a small molecule targeted for CDKL5. And he is also the co-founder of Child's Cure Genetic Research to support and fund research for rare pediatric genetic conditions. And he has also founded CDKL5 South Asia to support families in Indian subcontinent and beyond, which is a very high unmet need. So with that, I will stop sharing my screen and hand it over to Janu, who hopefully is there to join. All right, perfect. Janu, you are still muted and your camera is off as well if you're able to turn those on. We apologize for the technical delays. This is what happens with virtual talks all the time. We can see you now, Janu,
Yes. Yeah,
but we
hi.
no longer share your screen if you're able to share that again.
I will do that in a second.
Okay, great.
Thank you so much, and hello everyone. Sorry for this technical glitch. Uh, as and thank you so much, Ali and Dr. Bailey, for this overview. It is very helpful to give a perspective of what is gene therapy and how should we be expecting the next steps. Um,
All right, we
but
see everything. Great.
okay. Let me just make sure I change the pointer so you can see where I am pointing. And okay, so we often see amazing stories in news of children hearing for the first time or walking again after gene therapy. And every time I see these stories, one of the questions that comes to my mind is, when will this day come for CDKL5? As Dr. Whaley mentioned, many of these gene therapy programs are already FDA approved or in clinical trials. So we asked ourselves the same question and decided to take out actions. After waiting a long time, we decided to start our own gene replacement therapy program. I'll be sharing this. Sorry. Okay, I'm not sure what happened there.
Are you, yeah, if you're able to reshare. We can see it, it's just not in presentation mode.
Okay, can you hear me?
Yep, we can hear you, and the presentation looks good.
Okay, thank you.
Hmm.
So as I was saying, after waiting a long time, we decided to start our own gene replacement therapy program. I'll be sharing this information in a way that parents can easily follow, just like it was explained to me. In the interest of time, I will only be able to show a small snapshot of a much larger data set, which will be published shortly. If you have any questions, please put them in the chat and we'll make sure to answer them. So we learned a lot from the earlier efforts like UX055 and PTC therapeutics. And based on that, we designed our two promoters. These promoters are like switches that control where the CDKL5 gene works. As Dr. Bailey explained, that CDKL5 functions are predominantly in the brain, and we are now learning that it plays a role in other organs of the body like the heart, kidneys, gut, stomach, etc. What if these functions aren't just needed in one or two cell types, but they are needed more broadly? Thus, we wanted the promoters that are known for long-term expression as well as wide range of uh, cell and tissue that are expressed in wide range of cells and tissues. With that in mind, we created two options. And these options are the UBC different with different promoters. The UBC promoter, which can work in many cell types across the body, uh, and another one using CBH promoter, which promotes long-term expression. We're using an AAV9 as the delivery vehicle, and it's like a safe taxi that carries the gene to the right places in the cerebrospinal fluid, the CSF. AAV9 has strong safety record. It's well tolerated and effective based on the results from trials of other neurological disorders like MPS1, MPS2, GAN, and CLN7, some of which that are already FDA approved. As you see in this image, our approach was to measure these cell lines in two types of in as you see in this image, our approach is to test it in two promoters options in cell lines, both in HEK cell lines as well as iPSC cell lines. HEKs are common lab cells, whereas these iPSCs are specifically derived from CDKL5 patients. One promoter clearly performed better than the other promoter, but we know that this is just one piece of the puzzle, and these results may differ in bigger animal models or in pigs or mice or even in humans. So knowing that, the next thing we did was tried to study the efficacy of these promoters in mice. We use CDKL5 knockout mice from Jackson Labs. These are well studied and show behaviors of symptoms similar to CDKL5 human patients. Knockout means that the CDKL5 gene on both the chromosomes are removed. Wild type is a healthy mice or healthy and they are used for comparison or as controls. CDKL5 also mostly affects females that who are heterozygous, one good gene and one mutated, but their symptoms vary due to X chromosome inactivation. These makes it very hard for them to be studied consistently. Now, nonetheless, we included them to ensure clinical translation. Male knockout mice show clear phenotype and repeatable symptoms that match human features, making them a great model for testing these therapies. As you see here, we treated two groups of mice and in the gene therapy program that could and the aim was to show that if it could even help at a later age in life. 6-week-old mice were treated, and 6 week old is roughly like a human adult. We ran various behavioral tests throughout. And then we measured the CDKL5 protein expression and EB2 levels in the brain. EB2 is a downstream phosphorylation target of CDKL5. As, as we see in these results, there was no weight difference across these groups, which shows that AAV treatment did not harm growth or overall health, a good sign of safety. Similarly, in hind limb clasping, which shows motor issues in CDKL5 patients, perhaps like fisted hands and curled toes that we see in our kids, this treatment rescued this phenotype, which is meaningful for potential human benefits. So as you see in this graph here, what we are expecting, these are the wild type mice that are the controls, and the expectation is that after treatment, these mice should behave as closely as possible to the wild type. And this is male and this is female group, and this is what we are actually seeing after treatment. Similarly, the real-time PCR system was employed to quantify the relative mRNA expression levels of the target genes. These measurements showed good CDKL5 levels in the key areas of the brain, like the hippocampus in both male and female mice. This also helped us pick vector 1 as the better option as compared to vector 2. We then tested this top vector in newborn, 1 day old mice, in CDKL5 mice at the Jackson labs. As you see over here in contextual fear conditioning test, where the mice learns to freeze at the sound linked to a mild shock. The treatment in this mice improved uh, improved the memory aspect. As we see here, this P1 ICV injected mice, our vector achieves high expression in the brain and relatively much lower expression in the liver. So what have we, along with these preclinical work, we've had the pre-IND discussion with the FDA. They reviewed the data and gave helpful feedback. We asked some of the questions that we asked in the pre-IND were like, if we could use wild-type pigs for toxicology studies instead of non-human primates like monkeys. And they agreed, the FDA agreed to that. These larger animals are needed as the brain sizes are comparable to humans. This also gives us coverage of two animal species, mice and pigs. They also shared thoughts on our CMC proposal and plan for the clinical and the clinical protocol synopsis feedback with dose escalation. Right now, what we are doing is the toxicology studies through March of 2026 with the National Swine Testing Center and National Swine Resource and Research Center at the University of Missouri using ICM delivery. These centers also have knockout pigs that should demonstrate spontaneous seizures and reduce post-birth survival, similar to what we see in CDKL5 patients. Using this will allow us to assess the therapeutic benefits on epilepsy as well as survival, while also informing on safety and enabling comparison back to the mice to our mice studies. Manufacturing or CMC is underway with our partners at Medica Bio in Texas. Once toxicology is completed, the next step is going to be IND submission. The IND application is a very important regulatory step to initiate first-in-human CDKL5 gene therapy clinical trial. It includes an overview of the therapy, details on the gene, the design, production, purity, potencies, stability, sterility, etc. And it also includes the preclinical and tox data, the clinical strategy on how it works in animals to support human dosing, the full phase 1 study design, the objectives, inclusion and exclusion criteria, safety and long-term monitoring, endpoints, etc. We've partnered up with UCSF. We plan a phase 1/2 clinical trial to check the safety and early efficacy using clinical biomarkers and long and short-term monitoring. Concurrently, we plan to start the approval with SIDSCO, the Indian Drug Approval Agency, under the guidances of Dr. Gulati Lingappa and Dr. Aripilara. These trials may run concurrently in India and the US at the same time. The timeline. Please take this timeline with a grain of salt. We want to underpromise and overdeliver. Our community has faced disappointments before with projects that started but never reached clinics. Maybe we haven't shown the urgency and severity of CDKL5 enough. This program is a chance for treatment and hope, but it depends on two key factors: fundraising and the IFDA approval. The IFDA has been supportive in providing support. Has been supportive with clear feedback, and us with the strong team, we are optimistic about reaching the clinic. Current steps include IND enabling toxicology studies, manufacturing and finalizing the clinical protocol and biomarkers. This should lead us to the IND submission and trial. The clinical journey, especially this gene therapy program, has been a true team effort, and we are deeply grateful to everyone who has been part of this. A special thank you to ASGCT for creating this incredible platform and these annual meetings that bring together the right people and the resources under one roof. Your support has made it possible for us to reach this point so quickly. We are happy to answer any questions you may have at this point.
Thank you. Wonderful presentations. I do see there's a few questions submitted.
So I think I will take the first one, Allie.
Yeah, go ahead.
I think that makes sense. So the question is gene therapy designed for a specific child/mutation, which is a great question. So gene therapies can be designed for specific variants or for a given patient. And generally, that is for gene editing approaches or using antisense oligonucleotides, depending on the strategy. In the case of this gene replacement therapy that Janu described and ones that are similar to it, it would be broadly applicable to patients that have loss of function mutations, which is my understanding is the majority of patients. For some CDKL5 patients, though, it's my understanding there may be some potential gain of function in terms of the toxicity coming from the variant. It is unknown at this time whether gene addition would be helpful for that patient or those patients, and that's where additional research would be needed. But as of right now, it should be broadly usable for the majority of patients. And then, Allie, if you want to maybe go through the questions, then you can read out, and then
Yeah.
you and I can always decide who might be the best one to answer that.
Well, I think an important one I do want to get to, I'm seeing a couple things about where's the best place to donate, what's the best way we can help. So I do want to make sure that gets addressed, especially for action items. If you have some specific action items for the community, it sounds like there's interest.
Yes, thank you so much for that. So we've put together the website cdkl5genetherapy.com, which talks about all the work that has been done, what are the next steps, what are the teams that are involved in each of these stages of work, and where you can donate, and what would be the expectations in terms of who are the inclusion exclusion criteria, any potential more updates that we will be giving in the near future. So that will be the source where all of this information can be found. Thank you.
It sounds like you do have collaborators like the Lulu Foundation. I
Yes,
know you,
so
Andy.
yeah, so we are working to collaborate with the Lulu Foundation, the Howard family, Dr. David and Catherine Frame, CDKL5 in color, Dr. Kristen Loomis, Ruti Modia. And we are also already in conversations with Dr. Heather Olson at Boston Children's Hospital, Dr. Tim Benke at Colorado Children's Hospital. Ultragenyx has been always very collaborative in terms of sharing information and expertise. We are learning a lot from the Fox G Foundation. Dr. Guy and Nasha are great sources, and they are doing a similar program for Fox G1. So we are asking a lot of questions back and forth with their teams, and and learning a lot from them. Christina, McLean, Dr. Yale, Dr. Yael, and Dr. Allison. Allison is leading the Angelman and the through her program called FAST. So she's leading the gene therapy work there. So in the disorder community, there aren't many unlimited resources. So we are collaborating with everyone and learning from one another, and and like trying to share as much as possible without having to reinvent the wheel.
I think that's really great. And I know you mentioned some really great other organizations doing a ton of great work in this space. So it's great to hear you're connected with them. Um, I am seeing some questions about, I think this is probably the most significant parent question anyone has, timeline. And I know it's also the hardest question to answer. I just want to give one or both of you the opportunity to shine a little bit more on timeline, as it is equally frustrating, uncertain, hopeful that things can move quickly. Um, but I think people just want a little context to this.
Hmm. So, Dr. Bailey, if you want to go first.
I think for this specific timeline, I think that would be better for you to speak to, and then I can always chime in at the end.
Yeah, sure. So, as I mentioned, that everything depends on two factors: is funding as well as the IND submission. So right now we have the pre-IND discussion with the FDA. They gave us a clear expectation what they want to see in the next steps. And the whole team was very pleasantly surprised how detailed that feedback was. So, and we are taking these actions, and we are doing all the work implementing those work. So, as we see here, in terms of biomarkers, we have some ideas on the biomarkers. We are with the partners, these labs are running these tests to establish these biomarkers and confirming them. Similarly, the endpoints of the studies of these studies, we are learning from other studies like Rett syndrome, and what the endpoints were there and how we can use them, and similarly tweak them for CDKL5. The CMC is happening in parallel right now as we talk. The toxicology study is happening right now as we talk, and the finding the CDKL5 knockout pig model was huge. So that gives us like ultra assurance in one more species in which we can see symptoms more closer to humans as compared to mice. So all this work is already undergoing and on the way. And as soon as all of this is done, we will submit the IND submission. And from there is the clinical trial at UCSF and Rainbow Children's Hospital in India. I think this was one of the questions that had come in early on, that, if my child is in Iran, then where would and how would this therapy look like? So that's why we are trying to do it concurrently with CIDSCO in India. That's the Indian FDA agency, if you will. And the aim is for the families or the parents who are not able to travel to the US, there will be another alternate in India. So this is all. There's a lot of work in the pipeline. There are many forthcoming steps, and this, as in when we move forward, I will be providing more details and more granularities on the timelines, and especially the inclusion and exclusion criteria too. There was one of the questions I remember now was like, my daughter is 29 years old. Will she be able to participate? Yes. So that was the whole reason why we tested this gene therapy in two age groups. We did 1 day old as well as 6 weeks old. So 6 weeks old is human equivalent to sexual maturity. So from that perspective, anybody 18 years and older will also could also participate in these trials. And obviously, with the expectations on the efficacy and the gains, we all know about the plasticity of the brain and the rewiring. So the gains in older children or older patients may not be as significant as we see in a younger child. So this obviously we all know this, that the sooner the treatment it's better, but nonetheless there is always uh, with CDKL5 being able to corrective from Zhou Zhao's paper publication that we had learned years ago, that even in older patients there is they can be treated, and CDKL5 can then help uh, with those rescue those phenotypes that come with this disorder.
So that was great. And I also just want to add on in terms of, trying to level set expectations. Even after FDA approval is received for a clinical trial to be able to move forward, there are additional steps beyond that that then take time. And part of that is called clinical start site, trial setup. It can take months for a site, the location where the drug will be given, to get their own internal approvals to be able to move forward with the clinical trial. And when you have a first in human drug, which this will be, meaning it has never been tested in a person and it's not FDA approved, trial enrollment is done on a rolling basis because safety is the first thing they look at. So that means one patient is dosed and then they will wait months to make sure it is well tolerated and safe before they dose the next patient. So I just want to say that because although a trial starts, it can then be slow for patients to get dosed as part of it because we do that to ensure the best safety we can. This is experimental and unknown. So even after a trial starts, it may take months for then patients to be enrolled. And then the cadence of that may become faster as the trial goes on and we see there is better, that it's being well tolerated and it's safe, but just to set expectations on that.
Thank you for that, Dr. Bailey. Yes. So in our protocol synopsis, we proposed a 3-month wait period between the first set of patients and then the second set of patients. So yeah, as Dr. Bailey mentioned, it's not going to be, uh, like next day everybody are getting treated. It is going to be little bit of observation on the first few patients before we get on to the next ones. And this is a single site phase 1 clinical trial at UCSF in the US. So from that perspective, we are already working on their IRB approvals.
And then speaking of potential opening of a clinical trial someday, do we know the best place that people could stay updated? Is there, we have clinicaltrials.gov. That's one spot you can go to see if a
Yeah.
clinical trial is open, but just if you wanted to make a recommendation or if they should hear from you directly.
Yeah, so
Um,
that information will be published on CDKL5 gene therapy,
okay.
clinicaltrials.gov, and UCSF website. So these are some of the resources, and uh, I am uh, uh, I am engaged in the patient community, so you
Hmm.
will see this on Facebook and Instagram as well.
Great. I hope everyone will stay tuned. And So we kind of went over some of the questions about age and timeline. I think you can also send out this recorded. You can send out this recording after to the community as well
Yeah.
as like
So
the link to that website so everyone has the same information. Are either of you, I don't know, Dr. Bailey, if you're able to speak to this. Someone did ask on if there's the effectiveness of gene therapy in Rett syndrome. I do know there are some clinical trials
Um,
underway for Rett if there's
yeah,
some
so there are
similarities.
there are a couple clinical trials that are underway. To my knowledge, they have not published any results, and so it is important to wait for the publication of results because those results get peer reviewed by clinicians, scientists, people who rigorously look at what is being said, to then have it reported in a way that is fair and accurate. I believe there have been press releases by companies. Keep in mind, these are generally very favorable. They must be to help sustain programs. So they sound very promising, but to my knowledge, we don't have actual results yet. And the reason for that is it often takes a few years to gather enough information to feel confident in what the results are before you disclose them to the public. And that's in part because patients are dosed on a rolling basis and, they get studied over time to see what effects are.
So what so as Dr. Bailey mentioned, there are actually three clinical trials in right now underway. The Tasha, the Neurogen, and then this one is for overexpression or duplication of the MECP2 gene. And they did have one severe adverse effect, which was noted in November last year, where dose escalation and then where you're waiting, that waiting period before you enroll more patients. So this is where these clinical trials for Red are right now. So we don't have the published data yet, but the higher one of the patients was treated with with a very high dose, which was apparently safe in their studies, but the patient didn't survive. So from that perspective, it you know like i- i- it is uh, it is very important to to ensure the safety toxicology studies are done in a proper manner. I'm not saying that they didn't do it in a proper manner, but each human body is different. So from that, there's always that sweet spot, the balance that we are keeping in mind, that we maintain, the team is keeping in mind, that like we can go up to higher doses, but uh, we don't want any adverse effects to happen.
And I know you've told me about this, Shania, but can you update? Someone did ask about the current status of Ultragenyx gene therapy program. I don't know if you wanted to
Uh,
provide a brief update on that as well.
Sure. There was a press release last month regarding the Ultragenyx program, which was shared by the foundations like US, Italy, and I think Canada, and perhaps one more. So, and they said that this program is still like Ultragenyx is still working on it, but they have I think four other programs which are in BLA state, meaning the IND discussion with the FDA. So they are prioritizing those programs before CDKL5, and once they go through success, then they will take up the CDKL5 project back on track. So we are hoping one day that our children or our patients will have many different options, and then we can choose from there.
Yes, that would be wonderful. Well, there's lots of great feedback, a lot of positive feedback in the chat. So thank you both. I don't know if you wanted to say any final sentiment before we close the webinar today.
Uh, Dr. Bailey, you want to go first?
I just want to say thank you so much for the opportunity to speak to you this morning and for your engagement. It seems like this is a highly engaged community that is passionate about trying to find treatments, and that is important for these types of therapies to move forward. You know, as much as we would like for industry to do the heavy lifting for us, it's not always feasible. And so it's going to take the whole village to really move this forward. So thank you all for what you do day in, day out for your children and your families, as well as for trying to help push all of this forward.
And I want to thank everybody who could join, as well as all the CDKL5 families, the ones who've already lost the loved ones while waiting for a cure, and the ones who are still going through the burden of this devastating disorder in hopes of a cure. So, please don't lose hope. We will get there very soon.
Thank you all.
Thank you. Thanks.
Have a wonderful rest of your day.
Thank you all. Bye.